All right, we can start. We previously finished with structure of proteins, I think, and then we zoomed in on the function of proteins as enzymes, and then I think previously we started to look at metabolism of proteins as the breakdown to look at anabolism, that is the buildup of proteins from smaller molecules, or let's say protein synthesis. So synthesis of proteins from amino acids. So that is anabolism of proteins. That's what we are doing today. And I would say that today's lecture is much more of connecting the dots I realized I went offline for some few seconds, so I stopped talking, waiting for reconnection before I continue. So as I was saying, I know that in first year, you did some protein synthesis. That is the process of transcription, translation. So we'll be connecting some dots. Perhaps in the first year, certain things were not so clear. This time around, second year, we will go a little bit down to the level of the amino acid to look at the synthesis build up of that knowledge. So it's not just, the building of the proteins, but even the building of the amino acids themselves before they even will continue to build the proteins. We looked at this previously when we were looking at catabolism of proteins, when we saw that proteins which are taken in diet are broken down into amino acids. And these amino acids undergo two major processes of catabolism, which are transamination and deamination. And then we saw the that's go. After deamination, you have the formation of urea, which is excreted in urine. After transamination, what is formed is a keto acid that goes into energy generation, can go into gluconeogenesis, into the formation of glucose, or into the synthesis of fats, and even into the synthesis of non-essential amino acids. So take note of this last point here. non-essential amino acids. That is synthesis. So that is anabolism. That's anabolism there. Then also we see right from here, the amino acid goes to protein synthesis. So that is also anabolism. And we see there is even synthesis of some nitrogenous compounds. So you have proteins, amino acids going even into the synthesis of compounds like the nitrogenous bases that you find in nucleic acids. So our focus is going to be on what you see, and then also down here you see synthesis of non-essential amino acids. So those are the sections that we are going to pay attention to. We are not looking at metabolism of other nitrogenous compounds, so we will not be considering this. We'll have to leave some of the things so that you'll be interested to come and do your BTech. When you come to do BTech, we'll continue to look at metabolism of nucleic acids. So there you will see the synthesis of the nitrogenous bases coming from the amino acid residues. Right. Now we'll start with some revisions. As I was saying from the beginning, you did some protein synthesis looking at what you refer to as the central dogma of molecular biology, and that is to say that from the genes in the DNA, you have a process of transcription, which is the copying of the codes in the genes onto a messenger RNA that's in a eukaryotic cell that will be occurring in a nucleus. And then the messenger RNA moves to the cytoplasm, where there is a process of translation. That is copied onto the messenger RNA, is being translated now to the corresponding peptide or the corresponding protein. And it is that protein that gives an ultimate biochemical response. Because we've seen earlier the functions of proteins, that proteins can be hormones, they can be neurotransmitters, and then we even zoomed in on a particular function of proteins as enzymes. So you see, if a biochemical reaction needs to be catalyzed and a gene is activated, what happens is that that gene is then translated into the protein, which is the enzyme that will then catalyze the biochemical reaction. So you remember that process which you studied in the first year. Then also you want to remind yourself of the genetic code, where you have triplets of nucleotides that are coding for particular amino acids. These you find on the messenger RNA, referred to as codons. And you also have the corresponding anticodon on the transfer RNA. You want to revise those things. And so you see from, say, the first column, first row, you have some genetic code which is coding for phenylalanine and leucine. Column, still the first row, you find that or the two codes there are for their purpose in termination of the process of translation. And then you also see the next, you have two codes coding for the amino acid cysteine. There too, you have a stop codon, and then you have a codon for tryptophan. So you can look at all of these genetic code, coding for the 20 of these codes, but you have 4 of them that are special. So you have what you refer to as the start codon, which is red, AUG. To remember it, just remember that like a short form of August is coding for methionine. And what it also means is that the synthesis of proteins, let's say in eukaryotes, is always starting with methionine, because of course in prokaryotes it's going to be a derivative of the methionine. We have the formyl methionine, but the codon is the same, AUG. Then you have 3 stop codons: uracil, adenine, uracil, adenine, guanine, uracil, guanine, adenine. These 3, they are stop codons. So there's no need cramping all of these ones. You can keep them in mind. This is easy to keep, methionine. And even the stop codons, they are also easy to keep in mind. For instance, if you look at this stop codon, UAG, UGA, the 2 of them is just the changing of position of the adenine and the guanine. Uracil there is constant. You have that stop codon. And then the other one is uracil, adenine. So those 3 stop codons are also easy to remember. So you have all of these genetic codes that are corresponding to particular amino acids. Out of the 20 amino acids we know to be the protein amino acids. Then you also want to remind yourself of this process of translation. So here you have, say, the beginning, initiation of the process is such that you have that's a transfer RNA. The red there is a transfer RNA with an anticodon, UAC, that is corresponding to the start codon, AUG, on the messenger RNA. And we see it's carrying methionine, the amino acid, the first amino acid is carrying there is methionine. It's bound to the small ribosomal subunit. And that complex will scan for the start codon. And once it gets to the start codon, it binds onto the start codon. It triggers or initiates the formation of the initiation complex, where you have the large ribosomal subunit coming to bind onto that transfer RNA and methionine together with a small ribosomal. We can zoom in on that initiation complex so you can see it much more, where you now see the various parts of the initiation complex, particularly what we refer to as the the EPA sites of the large ribosomal subunit. So what I mean, so you have this large ribosomal subunit, and inside it you have 3 sites that are labeled E, P, A. I mean, E refers to exit. That is where a transfer RNA that brings an amino acid to the complex to bind into the peptide will exit the complex. And then you have the P. P means peptidyl. It is there that you find the peptide that is being formed. Is site that the next amino acid which is coming to the complex will be coming to. So what you see here is that you have a transfer RNA. So transfer RNA carrying the amino acid phenylalanine, but already you have a dipeptide formed. So the methionine, which was the first, is bound to tryptophan, and you see phenylalanine coming to the A site, the amino acid site. Okay, somebody is saying, okay, it's a different message altogether. All right. Okay. So then you have the transfer RNA bringing in phenylalanine. So what it means is that phenylalanine, once it comes, this tryptophan will then come to bind onto it. So you see all the parts, the anticodon, which is from the transfer RNA, the codon, which is on the messenger RNA, the small ribosomal subunit, and the large ribosomal subunit. So this whole thing is what is being referred to as the initiation complex. So you want to pay attention to the sites of the initiation complex, EPA sites. At the end of the lecture, I'll actually send you an animation so you can watch the process of translation, or even transcription, translation. You can watch it as a video, because some of these things, I know how it is when you are looking on it on paper and it's being explained by someone. It can be difficult for you to understand, even if you take the textbook looking into it. But if you watch some animation of the process, it will make it much easier for you. You can use it to, study. Then, of course, that process that we're referring to keeps on until you have a long peptide chain, and that's we refer to as elongation. So if you have transfer RNA, let's say that is the beginning, where you have phenylalanine on a transfer RNA on the start codon, and then you have transfer RNA carrying the next amino acid coming to the A site, and then you see the this phenylalanine that is, the arrow showing is coming to bind onto the amino acid that has just come to the A site. And once that binds to the A site, you now have a dipeptide which is formed. And the next thing you see is the process of translocation. There will be a shift of the complex, the ribosomal complex, to the right. And you see that dipeptide, the transfer RNA that is carrying the dipeptide, has now moved into the P site. You can see here it's in the A site, but here it has moved to the P site. And what it means is that the transfer RNA that was carrying that first, phenylalanine here in the P site shifts also to the E site and leaves. So because that's the exit site and leaves. So it leaves the complex. It can go and pick another amino acid and come back. The process continues like that. So that cycle continues until you have the whole peptide or the protein formed. And that protein can undergo some form of processing to become what it should be, whether it should be a hormone or a neurotransmitter or it's an enzyme to carry out its function. So that process continues until it reaches the stop codon and ends there. So yes, we have reached the termination where you reach a stop codon. So when you reach a stop codon, when the complex reaches a stop codon, that stop codon does not code for any amino acid. And so in the end, you have what? A release factor. A release factor is a protein, actually. It's a protein. So you have some release factors that bind into the A site when it reaches the stop codon. And once it binds to the A site, it triggers the hydrolysis of the peptide away from the transfer RNA, the last transfer RNA, which is carrying the peptide. And then you have the peptide free. And then in the end, you also have the dissociation of the whole complex. So that will be the end, the termination of the process of translation. All right. So you want to remind yourself of this. I know you did it in first year, so this is some form of revision. Again, even this one, we saw it again this semester, the amino acids. The amino acids. That we have, what I want by the body, and so it will have to be consumed. And these essential amino acids are the ones that are, in the dash lines. They are circled in dash lines. And the non-essential are the ones that the body can synthesize. So like I was saying that because you already did protein synthesis, what we just revised, we want to take a look at the synthesis of the amino acids themselves. And so if we want to take a look at synthesis of amino acids, then of course, we'll be looking at the synthesis of mostly the non-essential amino acids, because those are the ones that are synthesized by the body. The rest, which are essential, they are consumed as dietary proteins. They are consumed in dietary proteins. So they become available to the body through food. So for instance, alanine, is in bold, and so that is non-essential. That means to be synthesized in the body. Glycine, yes. Proline, tyrosine, aspartic acid, glutamic acid, arginine. Arginine, it can be essential, it can be non-essential. At developmental stages, in our developmental stages as children, so we are not able to synthesize arginine until at a certain age we begin to can be essential. We have serine. Serine being essential, being non-essential. We have cysteine also being non-essential. Methionine, no, methionine is not. But except that cysteine, even methionine, which is being shown here as essential, it can also be non-essential at certain points. If at a point it is needed in large amounts, such that you can have the synthesis of cysteine from it. That is if the body is not able to synthesize cysteine, or cysteine is needed in a certain large amounts at a certain point. Methionine can become non-essential, such that you have to synthesize some more of it there. We have asparagine, glutamine also being non-essential. So what we'll do is that we'll take some of these non-essential amino acids and take a look at their biosynthesis. Because if you want to take a look at all these amino acids and their biosynthesis, usually you come to see, sometimes if you go to see, the metabolic pathway is so complex, like, because everything is interconnected. I mentioned it before that metabolism of these biomolecules is not like it's a standalone. So you'll see very soon that you have metabolism of proteins connected with metabolism of carbohydrates. I think you even saw some earlier. And then you see there's even a connection with lipids. Even looking at the very first slide we looked at, you could see there was some formation of keto acids, which will go into formation of fatty acids, or lipids. And then you see some gluconeogenesis, synthesis of glucose. So that's already telling you that there is that interconnection between the metabolism of proteins and carbohydrates and lipids there. Even you saw there's formation of other nitrogenous compounds. And I mentioned that some of that is metabolism of nitrogenous bases that you find in nucleotides. So that is already going into metabolism of nucleic acids, DNA and RNA. So the interconnection is there as well. So let's remind ourselves again with one thing, like just to mention, the various compounds along the line, the intermediates that are formed in the process of glycolysis, Krebs cycle, because you did that. And you'll be seeing those intermediates that you saw in the process of glycolysis and Krebs cycle, coming into the synthesis of certain amino acids. So just to quickly look over it, when you are studying, go back, because I told you from the beginning, from the first year, and now you are combining the things to look at metabolism of proteins, but it's not on its own. It's connected to metabolism of carbohydrates. So you'll be looking at certain compounds that are taking part in the reactions, and some of these compounds you'll find them as intermediates in process of glycolysis, or in the TCA cycle. So quickly, glycolysis, you saw. If what you refer to as maybe investment phase, payoff phase, but you can just say phase 1, phase 2. The first phase, which is here, is represented as the first phase is at the top, and then the second phase is below. So as you can see, the arrow goes, that way, which means the beginning is from glucose and it ends at pyruvate. So you see from the beginning, glucose, and some phosphorylation process occurs. You have glucose 6-phosphate. Then you have some isomerization. You have fructose 6-phosphate. Glycolysis, breaking down. So then you now have 2 molecules, glyceraldehyde, 3-phosphate, and then dihydroxyacetone phosphate. Further, you then will have another phosphorylation. Of course, you'll be having these dihydroxyacetone phosphates, getting converted also to form the glyceraldehyde phosphate. That's why you're having the arrow showing in 2 directions. You have 1,3 phosphoglycerate. Then you have some dephosphorylation occurring. You have 3 phosphoglycerate. Isomerization again, 2 phosphoglycerate. And then further, you have some phosphoenol pyruvate. And in the last end, you'll find pyruvate. Intentionally, I'm just mentioning the names of the compounds so that as we continue, you begin to see those compounds that you saw in glycolysis. Again, you'll be seeing compounds that you saw in the Krebs cycle, those intermediates, also in the synthesis of amino acids. So for instance, you have the acetyl-CoA, which will be coming from pyruvate from glycolysis. And the first thing is there's that binding with oxaloacetate. You have a citric acid. And this is why sometimes it's referred to as the citric acid cycle or the tricarboxylic acid cycle, because this, the beginning, what you see here, citric acid, isocitric acid, these are tricarboxylic acids. They have 3 carboxylic acid groups in them. That's why sometimes it's referred to either citric acid cycle or tricarboxylic acid cycle. Then you have the alpha-ketoglutarate acid, which is also either it is alpha-ketoglutarate or alpha-ketogluteric acid. It is the same thing. So, for instance, you see in some books, when you are reading, you see like citric acid, they will just write citrate. Is the same. If you see isocitrate, is the same as isocitric acid. You saw something like succinyl-CoA, succinate. These 2 you can see, succinic acid, fumarate, fumaric acid, malate, malic acid, oxaloacetate, oxaloacetic acid. So these names, when these things that you saw in the Krebs cycle. Now, from there, you now see if you are looking at say the overview, the general view of the synthesis of the amino acids. You see, we were looking at the sum of these amino acids which are non-essential, which means the body will synthesize them. Of course, the body synthesizes not only the non-essential amino acids. There are some of those amino acids we refer to as essential amino acids that the body synthesizes them as well, except that not in such adequate amounts, so that there has to be some compensation synthesized. So for those ones, they are referred to as essential amino acids. But it doesn't mean that they don't get synthesized at all in the body. So when you look at the process of glycolysis, which is central, so from glucose coming, the key there is showing glycolysis in blue. And we have a pentose phosphate pathway, which is another pathway in carbohydrate metabolism, which goes into the synthesis of nucleic acids. Then you have the citric acid cycle. That is the Krebs cycle, or the tricarboxylic acid cycle, which we just saw. And you see that there is the synthesis of amino acids that are connected to these processes. So you see glucose 6-phosphate coming down, 3-phosphoglycerate, phosphoenol pyruvate. So that is the glycolytic pathway. Then that joins the citric acid cycle. Glucose 6-phosphate may go into the pentose phosphate pathway, sometimes referred to as the hexose monophosphate shunt. And that pathway can go into the synthesis of amino acids such as histidine and serine. So you can see the connection. It's carbohydrate metabolism that is connected to amino acid metabolism, amino acid synthesis. So that is protein metabolism. Then we see to the synthesis of serine. So that's another amino acid. It may even go into another path where you have the formation of phenylalanine, tyrosine, tryptophan. When we classify these amino acids, what are they? These are the aromatic amino acids with the aromatic side chain. Then you see pyruvate, along that line, pyruvate can also go into the synthesis of amino acids such as alanine, valine, isoleucine, and leucine. The citric acid cycle itself, you see oxaloacetate can go into the synthesis of aspartate or aspartic acid, asparagine, methionine, threonine, and lysine. Alpha ketoglutarate can also go into the synthesis of glutamate or glutamic acid, glutamine, proline, and arginine. So you see these amino acids, their biosynthesis is connected to the carbohydrate metabolism pathways. So that's just an overview of the biosynthesis of these amino acids. So what we'll do now is we'll take specific amino acids, different ones, and begin to look at the different pathways in which they get metabolized. Of course, if you want to look at the pathways all together interconnected, it will be so clumsy. So what we try to do is, as much as possible, we take one amino acid at a time and look at the pathway that it is synthesized in. So you can appreciate the biosynthetic pathway, or let's say the anabolic pathway of that amino acid. it doesn't mean that the pathway is standing alone. If you want to take it as a whole, like I said, it will be clumsy. There will be lots of pathways that will be joined, and it will just be so clumsy that you will not see the essence of it. But we'll take it one after the other. So let's take alanine. So what do we see? Here too, we see, glycolytic pathway is still central here. So we see glucose undergoing glycolysis. And what you find, the product of glycolysis there is pyruvate. And that pyruvate goes into the formation of alanine through the process of transamination. So you have an amine group from an amino acid, usually glutamate, that gets transferred onto pyruvate, and you have the formation of alanine. That is occurring in the muscle. Of course, glycolysis, after you have glucose in the bloodstream going to the muscle tissues, it is in the muscle tissues that you have the process, this glycolysis, generating energy, for the body. And glycolytic process, it can go into the formation of alanine. That alanine, after it is formed, of course, it goes into the bloodstream, moves to the liver, and in the liver, it can then undergo breakdown, what we saw much earlier, the process of deamination. So the amine group is removed from the alanine, and that's amine group. And then there's that formation of urea in that process, whilst you have the carbon chain back as pyruvate. So the process continues, and that pyruvate can go back through the process of gluconeogenesis, generating back glucose molecules. Of course, the right pathway is purely carbohydrate metabolism. This in an anaerobic condition where you have lactic acid or lactate being formed. That's you see what we refer to as lactic acidosis. That is that process that is being shown over there. All right. Then we can take aspartate and asparagine, or aspartic acid and asparagine. So the two of them, they are being formed in, same pathway. And right at the beginning, what do we see? Oxaloacetate. Oxaloacetate. We saw oxaloacetate in the Krebs cycle. The very first reaction where you see the acetyl-CoA coming into the Krebs cycle, it binds to oxaloacetate to form citric acid. So oxaloacetate is there from the Krebs cycle. So that is an amino acid. Glutamate is transferring. So you have a transfer of this amine group. We saw this type of reaction when we were looking at the classes of enzymes. We refer to transferases, which are responsible for the transfer of functional groups from one molecule to the other. And we see here, if you take a close look at the structure of oxaloacetate, you see there is a double bond O here in the middle. In place of that, in aspartate, you see there's an amine group in place of that double bond O in aspartate. That amine group is coming from glutamate. That amine group is coming from glutamate, being transferred to that position in oxaloacetate. And if you pay attention, you see that after it transfers that amine group, it forms alpha ketoglutarate. And look at this. In place of the amine group in glutamate, you now have double bond O. Where is that double bond O coming from? The double bond O is coming from the oxaloacetate. So there is that exchange between oxaloacetate and glutamate, where you have glutamate giving out amine group and oxaloacetate giving out the double bond O to that position to form the alpha ketoglutarate. So you have aspartate synthesized in that process. The enzyme transaminase. Now, the aspartic acid can get further converted. From aspartic acid, you can have the synthesis of asparagine. You can have the synthesis of asparagine. Here too, if you pay close attention, look at this. Aspartic acid, you see the very first carboxylic acid group. You know, the O minus there can be written, in a molecular form as OH. But this is written like in an ionic form to show it is giving out a hydrogen ion. So you see, that is like a hydroxyl group which should be there in you see an amine group. In that position, you see an amine group. Where is the amine group coming from? See, the amine group is coming from glutamine. So, but this time around, it is this NH2 in glutamine. So you see, as it forms glutamate, in place of that NH2, you have the O minus. So the same thing is occurring, that, sort of the exchange. So you have the amine group coming to the position of that, O minus or a hydroxyl group in aspartic acid, whilst you have that group, the O minus or the hydroxyl group, going to that position where the NH2 is in glutamine to now form glutamate. So now you then have asparagine formed. So these are two amino acids which are being formed in that pathway, aspartic acid and asparagine. Right. We can also take a look at, say, the biosynthesis of, glutamine itself, glutamine. So glutamine is being synthesized from glutamate. And we see from the beginning, what you see is phosphorylation of glutamate. I mentioned before that when you look at many biochemical reaction pathways, they start with a process of phosphorylation. It's a process of activation of molecules to go down a certain, biochemical pathway. So you see ATP, it's a cellular form of energy in that reaction, giving out a phosphate group, and itself then becomes a diphosphate. So once it gives out one phosphate, ATP, then it becomes ADP, a diphosphate. Adenosine triphosphate gives out phosphate group, becomes where the O minus is, should be some hydroxyl group there. In place of that, you have a phosphate group. That phosphate group is coming from ATP, but that's an intermediate, acyl phosphate intermediate. Then in that position, an amine group coming from ammonia now comes to displace that phosphate group. And so you have NH2. You now have glutamine synthesized in that process. So you can have glutamine synthesized in that manner, where ammonia brings in the amine group onto glutamate to form glutamine. Can take a look at another one, serine. Serine. The beginning of the reaction. Look at it. 3-phosphoglycerate. Where did you see that? So in the glycolytic pathway, one of the intermediates was that 3-phosphoglycerate. And you see, the first thing which is happening here is some, form of oxidation. Some oxidation reaction is occurring. If you pay attention, you see that in place of this hydroxyl group, in 3-phosphoglycerate, you now have a double bond O. So there's some oxidation reaction occurring there. What you see there is this number here, like enzyme commission number. It's a form of international numbering of enzymes. You don't have to cramp it in mind. If you see the number, sometimes it's even written on some food products and things like that. You can just check online. You see which enzymes fall within this category. It can be different enzymes depending on which organism it is. It's actually dependent on the type of reaction that the enzyme is catalyzing. But the very first number that you can relate to quickly, when we did enzyme classification, we saw the first class of enzymes was oxidoreductases, enzymes which are involved in the catalysis of oxidation and reduction reactions, what you commonly refer to as redox reactions. That's number one, before all other things. So, and again, you even see the process which is occurring is an oxidation reaction. So that enzyme, that first number will be 1. So if you follow that, after oxidoreductases, the next was transferases. So you see, the next enzyme that you see here, it starts EC2, 2 point something. So it means the enzyme which is acting in this process is a transferase. The other numbers, of course, you cannot cramp them so much. If you work with this for some time, you'll get to know them. But as for the first numbers, you can easily relate. But to know the specific enzymes that can fall within this category, you just do a reference and you see. And here you see, again, glutamate. Glutamate is, giving that amine group. So, the enzyme is a transferase, so it's transferring amine group from glutamate. And where is it taking it to? You see this double bond O, that position where the hydroxyl group was, and it got oxidized. You now have in that position NH2. NH2. So the enzyme transferred the amine group from glutamate to that position, which means that double bond O will also go onto the glutamate, and now you have that oxoglutarate formed in the end. But all of these we are talking about, they are still intermediates. The product is serine. The product is serine. Now, again, if you look at a classification of enzymes that we saw, what was the next after transferases were hydrolases. Hydrolases. So this enzyme is a hydrolase. So you see, even that is water coming into that reaction, water. So it's a hydrolytic reaction, or what you say hydrolysis. And what is it hydrolyzing? It is hydrolyzing the phosphate group. So the phosphate group, is hydrolyzed away from the molecule. In place of that, you have a hydroxyl group. So you now have serine, the product. So that is synthesis of serine also there for you. Right. Now we can take synthesis of cysteine, one of the amino acids containing sulfur. There are two, cysteine and methionine in proteins. Of course, you have homocysteine, and I mentioned, there are some proteins you come across. Some amino acids you come across. They are there, but they are not found in proteins. They are not peptide amino acids. So they are involved in different processes, like what we are seeing here, homocysteine involved in the synthesis of cysteine. So out of it, you have cysteine being synthesized. We just saw the synthesis of serine. Now you see serine is reacting with homocysteine to give you the final product that we are looking at is cysteine. And how does that happen? In the first place, you have a dehydration reaction occurring in the first place. So you see that hydroxyl group is gone. This hydrogen is gone. So have this sulfur in the middle. So you now have that conjugation between homocysteine and serine. So you now have sister thionine. From that sister thionine, you then have the process of, again, hydrolysis. See, water comes in that reaction. And there is also some deamination occurring at the same time. So what you see is that you see alpha ketobutyrate. Here, what do you see? In place of that NH2, you are now seeing double bond O in the alpha ketobutyrate. And the end there is a methyl group, CH3. And you see the sulfur is what you now find in that sulfur hydral group in cysteine. That NH2 you see in the intermediate is still there, NH2. So it is the NH2 which is in that, from the homocysteine side, that is what is given out. That's in place of that is what you have the double bond O to have the alpha keto. Cysteine. Okay, so you've been talking about breakdown of amino acids, or even from secondary school, we've been talking about breakdown of know that proteins are not only broken down, but amino acids can also bind to form proteins. And which process? By which process will you have amino acids binding together to form proteins? That's what we looked at. The process of transcription, translation. You have the amino acids binding to give you the proteins. Okay. How about the amino acids themselves? The ones that we say they are non-essential, meaning that the body can synthesize them. How does the body synthesize those amino acids? So these are the things that we are looking at so that you can relate, practically with these things. Then we take tyrosine. Tyrosine. So you remember the structure of tyrosine is also one of these on the aromatic side chain. That's the difference it has with phenylalanine. So you see phenylalanine is here. Phenylalanine is having the 6-sided ring, the benzene ring. And you see tyrosine is having that hydroxyl group on it. So you can have a synthesis of tyrosine directly from phenylalanine, just by hydroxylation. And of course, that tyrosine, even though we are not looking at all these others, you can relate with it that you are just having further hydroxylation. So you see, you are now having a dihydroxy, phenylalanine. And that dihydroxyphenylalanine then goes into the synthesis of other compounds. So you see that biosynthesis of amino acids going into different pathways. All right, I've been talking for 1 hour, so I want to stop talking and now listen to you. The floor is open. If you have any questions, any comments, any contributions, anything you want to share with us, the floor is open.